Hello and welcome to the joint FKMCD Oxitec Public Educational Webinar, the 13th in our series. Today, we're going to talk about our Mosquito project, which we are launching this week, and what Florida Keys residents can expect in the coming weeks and months. We're joined today by Andrea Leal, the Director of the Florida Keys Mosquito Control District, and Nathan Rose, our Head of Regulatory Affairs at Oxitec. I'm Rajiv Vaidyanathan. I'm the Director of U.S. Programs at Oxitec, and I'll be moderating the discussion. At the end of this presentation, we will be answering questions which we are collating through the chat online. A word about these public educational webinars. Our joint webinars are intended to share information with residents of the Florida Keys and provide a forum to answer your questions. Our webinars are open to everyone. They are recorded and they are made available to everyone after the event. All of your questions relating to the webinar topic will be answered. Now, we might bunch some of them together because some of those questions might be similar topics. And if time runs out, we will continue accepting questions in writing. Please submit them to florida@oxitec.com. The questions and answers will be published in writing after the event with external or related online resources. Keep in mind that we will prioritize the questions which are immediately pertinent to the topics that Andrea and Nathan will cover. Welcome to our webinar. In today's agenda, we're going to talk about the deliverables of our Mosquito project, how the success of our project will be measured, and what to expect now and through the summer. Please ask your questions in the chat box, and we will defer to Andrea and Nathan at the end of this presentation. Andrea, please begin, and I will mute myself.
Thank you, Rajiv. We first started looking at this technology about a decade ago. As you can see by the graph at the top, right around 2009 and 2010, we had just under 100 cases of confirmed locally acquired dengue fever in Key West. At the time, we spent millions of dollars combating this outbreak, and unfortunately, we saw another outbreak again just this last year in Key Largo. So overall, we are seeing mosquito numbers for this particular vector above transmission thresholds. And we're talking about one mosquito. This mosquito is called Aedes aegypti and is responsible for transmitting diseases such as dengue fever, Zika, chikungunya, and yellow fever. The Aedes aegypti mosquito itself makes up a very small portion of our mosquito population, but is responsible for 100% of our human health risk. One of the other things about Aedes aegypti specifically is that we are seeing the beginnings of resistance in our local population, particularly to adult insecticides that we're using. So unfortunately, what we're seeing is over time, our control methods are shrinking. The number of control methods that we have available to us are shrinking. And that's really why we started looking at these novel technologies. Basically, we need more tools in our toolbox. And I think one of the great things about this technology particularly is that it is specific to Aedes aegypti mosquitoes only. So there'll be male releases, they'll seek out females, and no other species of mosquito, insect, etc. will be targeted by this. Which is extremely important for us here in the Florida Keys because we live in such a sensitive ecosystem. We're surrounded by a national marine sanctuary, and we want to make sure at Mosquito Control that we're doing our best job to protect our sensitive environment. Nathan, over to you.
Thank you, Andrea. What I'd like to do is just briefly recap what this technology is, and then we'll talk a little bit more about what the project is going to look like in the coming weeks in the Florida Keys. So for those who might be joining us for the first time, Oxitec and FKMCD together will be releasing a strain of Aedes aegypti called OX5034. And this is basically a strain of Aedes aegypti, which has had 2 genes inserted into these mosquitoes. One of them is a self-limiting gene, which causes the death of all of the female offspring of the male mosquitoes which we release. We only release male mosquitoes which cannot bite and cannot transmit disease. And then the second gene is a fluorescent marker gene, and you can see some pictures in the bottom right of what that looks like. This allows us to trace these mosquitoes in the field at any life stage. So male mosquitoes only are released, and what they do is they will fly out and they will go and mate with wild invasive female Aedes aegypti mosquitoes, and those females will then lay eggs. And when those eggs hatch, any females that hatch that have these genes in them will die in the early larval stages of development, while males that hatch will be able to carry that gene and pass on a copy of the gene again to their offspring. And so this gives targeted suppression of just the single species, Aedes aegypti, which, as Andrea said, is the main disease vector in the Florida Keys. It is safe, it's non-toxic, it's non-allergenic, and it also has proven effectiveness. So these mosquitoes have been released for several years in Brazil by Oxitec, where we've seen very effective suppression of the wild invasive Aedes aegypti population in cities where we've released these mosquitoes. And the way that these are released is the small box which you can see on the lower left part of this slide. This is a mosquito release box, and what this basically contains is the mosquito eggs, to which we add water and some diet. And what happens is that a few days after we've actually put these eggs into this box, and this box is in the environment, we start to see male OX5034 mosquitoes emerging from these boxes. So it's a very low tech, very simple way to actually release these mosquitoes in the environment. So the project that we're carrying out in Florida is what we call a regulatory pilot project. And I just want to contextualize this here so that it's a bit clearer as to what you can expect. A regulatory pilot project is a small project with high statistical power. That means that we have replication. We will release and basically carry out the same kind of project in multiple locations. And that allows us to actually carry out statistics to show whether the effect is significant or not. It uses a protocol that's approved by regulators, in this case by the Environmental Protection Agency and also by the Florida Department of Agriculture and Consumer Services. And this is really focused on understanding more about the biology of the mosquitoes which are released in the particular local environment, and then also about measuring the efficacy. So how effective are these in actually killing those female offspring? And based on all of the data from the lab and also from multiple years of releases in Brazil, we know that these mosquitoes kill the genes that these mosquitoes carry kill all of their female offspring. Another kind of project which would be carried out maybe after a regulatory pilot, and this is what we've done in Brazil, for example, would be a larger demonstration, and that's really to demonstrate an area-wide performance of a product like these mosquitoes. That would also be designed with the regulator and would also have comparisons with controls. And then finally, operational deployment, that's something which would likely come after a commercial registration of a product, is when this is actually in the hands of vector control authorities like the mosquito control districts to suppress vector populations over a much larger area. So here we're really just talking about the first of these, and that's a regulatory pilot project. So this slide here is just giving an outline of how we'll be measuring performance during these mosquito releases in the Florida Keys. The first thing to say is that this mosquito performs like a larvicide, which is specific to just the Aedes aegypti mosquitoes. It only kills the female larvae of the next generation. And some of the metrics that we'll be looking at throughout this project are things like the abundance, and this is where we actually look at the numbers of wild Aedes aegypti in traps in the project areas. And that allows us to look at the baseline population levels before we release any mosquitoes, and then also any changes in those populations of the wild Aedes aegypti over the course of the mosquito releases. We look at something called the overflooding ratio, and that's the ratio of the released Oxitec male mosquitoes to the number of wild males that are in the environment. And that's important because it helps us to achieve and measure the optimal dose rate. We can also measure the mating fraction, and that's the proportion of wild females that have been mated by the released Oxitec male mosquitoes. And that helps us to evaluate the proportion of the wild population which has actually been treated by these mosquitoes. And then finally, efficacy. And efficacy is the percentage of the treated females, that's the female offspring, that actually die as a result of the genes which they inherit. And here we're looking to confirm the 100% effectiveness against the treated females that we've already demonstrated in other places. And the way that data is collected throughout this project is really from a number of different traps, ways that we can catch mosquitoes in the field. And these are standard mosquito traps, which are used by mosquito control authorities throughout the country and also throughout the world to monitor wild mosquito populations. And the first of these is what's called an ovitrap or an egg trap. And that really allows the scientists to collect mosquito eggs in the field. And what this consists of is a small plastic cup that has a little wooden paddle on it. And that's a perfect environment for a female Aedes aegypti mosquito to lay her eggs. And then what we can do is maybe once a week, we'll go out, we'll collect all of those wooden paddles, take them back to the lab where we can then hatch those eggs and analyze all of the mosquitoes that hatch from those eggs. So we can look at what species they are. We can determine whether they're male or female. We can look in this case as to whether they carry this fluorescent marker gene or not. And then we can also confirm that all of the females that carry that fluorescent marker gene are dying as expected. The second kind of trap is an adult mosquito trap. And there are lots of different kinds of these, but the one that we'll be using throughout this project is called a BG Sentinel trap. And that has an attractant in it, which helps to attract adult mosquitoes into a catch bag, which can then again be collected once a week or so, taken back to the lab, and then those adult mosquitoes analyzed again to look at what species they are, to look again if they're male or female, and to look for that fluorescent marker as well. So both of those kind of traps will be placed throughout the project areas in the Florida Keys. And the samples that are collected from those throughout the project will be brought back to the labs, where they'll be analyzed in the ways that I've just described. So the purpose of this project is really to help to broaden the toolbox, as Andrea said, that is available to mosquito control authorities to protect communities against invasive species like Aedes aegypti, which has only been in this environment for a few hundred years. It's also important that this project is preserving the quality of life for residents and also the delicate ecosystem in the Florida Keys. And really the main aim here is to evaluate this innovative tool for fighting Aedes aegypti. And there are several components to this project. The first is community engagement, which has been going on for a long time in the Florida Keys, and which will continue throughout the actual mosquito releases to help the communities understand what the project is, what will be happening, what they can expect. The first phase of this project, which begins this week, is what we call Project A, and I'll talk a little bit more about that in one of the coming slides. And then a second phase of this project is Project B, where we release these mosquitoes over slightly larger areas. So this slide gives you a snapshot of what Project A and Project B are really about. Project A is where we have these mosquito release boxes releasing mosquitoes from single points, single locations in the Florida Keys. So we have 6 small areas which we have selected, and they will have regular placements of these devices over about 12 weeks. And over all of those areas, we'll be having around 12,000 of these non-biting male mosquitoes coming out per week across all of these areas in total. These locations for Project A, we have 1 on Ramrod Key, 2 on Cudjoe Key, and 3 on Vaca Key. And throughout those areas, as I've mentioned, we'll also have these egg traps and also these adult mosquito traps. Project B, which is the second phase of this project, will have multiple release points throughout small neighborhoods. And that'll see a small number of devices placed per week in up to 6 areas, and that might run for slightly longer, maybe about 16 weeks. And after both of these projects end, there will also be post-release monitoring to monitor the disappearance of these mosquitoes and their genes from the environment. So Project A, what's actually happening this week and in the coming weeks in the Florida Keys? On the right-hand side, you can see an illustrative map. This is not actually from the Florida Keys, but it's just here to show you what these projects will look like. And so in the center, you have a mosquito release box placed. And then throughout an area up to about 400 meters away from that central location, we will have both these adult traps and these egg traps placed. Quite a lot of them. We're looking at several dozen traps throughout these areas. And the Aedes aegypti mosquito will typically fly maybe 50 to 100 meters during its lifetime. So the mosquitoes that actually come out of these boxes are expected to stay within that sort of 50 to 100 meter radius that you can see there in the solid yellow circle. But we will continue to trap for a larger area around those release points up to 400 meters to be able to detect any mosquitoes that might actually be flying a little bit further. When we've released these mosquitoes in Brazil previously, we've seen that the average distance that they travel is about 50 meters during their lifetime. So during this project, we'll have these mosquito boxes placed in yards. And after about 2 weeks, after they've been placed, the eggs have hatched, those have developed into these male mosquitoes, you'll actually start to see those male mosquitoes coming out of those boxes. And they will emerge gradually. It won't all be on a single day, but they will then emerge over the next couple of weeks. And after 28 days, that box is then replaced with a new one. And so we'll be cycling these boxes in these locations to make sure that we have a continual release of these male mosquitoes throughout the project period. And throughout that period, the project team, FKMCD and Oxitec staff will be continually monitoring the adult and the egg traps around these release sites, taking them back to the lab and then analyzing what they're finding there. And so this first part of the project is focusing on a few different elements which we'll be evaluating. So we want to look at the duration of the effect. How long are these mosquitoes actually present in the environment? We want to look at the male flight range and the longevity or the lifespan of these mosquitoes. How long do they actually live in the field once they've come out of these boxes? We'll also be confirming the percentage kill of the female mosquito offspring. And that's really done through these ovitraps where we bring those eggs back into the lab, hatch them, and then look at the fluorescent larvae to confirm that the females are dying and that the males are surviving. We're also looking at the percentage of the wild population which is treated. That's the mating fraction, which I referred to earlier. And because this is a regulatory pilot trial, we'll be doing this in replication. So we have 6 different release sites. We also have 3 control sites where we're not actually releasing any mosquitoes, but where we will also be monitoring the wild mosquito population. So then we can compare what happens in a release site to one of these control sites with replication as well. And then throughout the summer, as I've said, Project A will last for about 12 weeks. Project B, which is the second phase, will place multiple boxes in small neighborhood release areas. And so this is also just an illustration. Again, this location is not actually in the Florida Keys in this picture, but it just shows you how we might place. Here we have about 10 different mosquito release boxes over a small neighborhood. And then interspersed between those are, again, the adult traps and the egg traps. And that, again, allows us to monitor what happens to the invasive Aedes aegypti population in that area throughout this project. After the releases end, so both for Project A and for Project B, the areas where the mosquitoes have been released will be monitored until we no longer see any of these fluorescent mosquitoes present in the environment. And we'll actually continue to monitor even after we stop seeing them. We'll continue to monitor for several weeks after that to confirm that they really have gone from the environment. And then following the end of the mosquito releases, the full data analysis should be completed and then shared with the regulators to help them to assess the performance of the mosquito in this context. So I'm going to hand back to Andrea here just to talk a little bit more about the ways in which we've been engaging the community in the run-up to this project.
Thank you, Nathan. As I mentioned before, we really have been looking at this at our district for about 10 years and have been talking about it ever since that time frame. We want to make sure that we're coordinating fully between ourselves and Oxitec and making sure that information is getting out to the public. We talk on the radio on a regular basis. We've been speaking with community groups and stakeholders throughout this entire process as well. And we've been going out and speaking with civic groups, just making sure that we're answering questions and engaging with folks out there. Our Board of Commissioners put on the ballot in 2016 a question to see what the support was for this project out there. And 31 of 33 of those voting precincts were in favor of moving forward with this particular project. So we've had some really great public support because of that. Next slide, please. And since we've announced that we're moving forward with this project particularly, we haven't slowed down. And in fact, we've really increased the amount of public education that we've been conducting. Unfortunately, COVID made that look a little different than normal. Instead of being able to be out and about in the community as much as we wanted to, we've had to go with virtual. And this webinar, including in one of those. This is our 13th public webinar, and I anticipate, continuing this type of community engagement in the future. Luckily, we're able to get out and about a little bit more than what we were last year. So I know that our public information officer, as well as Oxitec representatives, have been speaking in schools, speaking at community events. We're trying to make sure that we're out there answering questions that anybody may have. So community engagement is a very important part of this. And one of the great things we've seen as this project has progressed, as we're going door to door, we are getting a lot of positive feedback. We've got a lot of public support wanting to participate in the project. So for those out there listening to this that are participating in the project by either hosting boxes or traps, I just want to say thank you very much. We really appreciate your support in all of this. Next slide, please. So just kind of to summarize again what our project timeline is, we started community engagement way back when and plan to do community engagement for this project throughout the entirety of both projects. As Nathan said, this was a big week for us. We're very excited to put our first eggs out in the field this week. And we anticipate these projects running through probably late October. What you'll see is box placement, mosquitoes emerging through mosquito season. And throughout that entire time, we're going to be monitoring and evaluating what's going on. And then once these releases are finished, we will continue monitoring for as long as it takes for those mosquitoes to leave our environment. Next slide. And now I'll go ahead and turn it over to Rajiv.
Thank you both. A final word here. If people are interested in hosting what we call an OV trap or an egg trap, or if they're interested in hosting one of the BG Sentinel traps, that is a monitoring device for adult Aedes aegypti, please contact us. You can request a trap, you can sign up for updates, and you can volunteer as a project ambassador. Questions have been collecting this afternoon, and if we run out of time tonight, you can still email them to florida@oxitec.com, and we'll attempt to answer your question if it is not included in the growing FAQ or the post-event summary that we publish. So at this point, the questions have been collated, and some of them have been kind of bunched into one. So we might start with maybe a very broad set of questions, either Andrea or Nathan. This is something that you can both respond to, and then maybe get into some of the more personal and detailed responses. So there seem to be a number of people who are asking, what will I notice if I live near a release location?
I can go with that one first. So once a box has actually been placed in a release location, it'll take a couple of weeks before any male mosquitoes start to emerge from that box. And then those males will emerge at their own leisure over the next couple of weeks after that. And then once we have those boxes actually being cycled, so one releasing males and then another one being placed to kind of pick up where the first one finishes, we should then have male mosquitoes being released continuously throughout those project areas. So if you live in an area really close to a release box, you might expect to see an increased number of male Aedes aegypti mosquitoes there. But it's really important to know that these males are not interested in biting humans. They don't actually have the mouth parts to bite anyone. All they're interested in is finding female mosquitoes to mate with. So you might see an increased number of mosquitoes, but there is absolutely no harm to humans as a result of that.
And on the mosquito control side, you really shouldn't notice any sort of difference whatsoever. Our plan is to continue our operations as normal, which of course we do our operations depending upon what we're finding in each of these areas. So you really aren't going to notice much of a difference whatsoever, either from the mosquitoes emerging from the boxes or with mosquito control operations in your area.
Thank you to both of you. Now, I know you have talked about this in the webinar. The question is coming up, so maybe it bears repeating and a little emphasis. First question, how long is this trial from end to end? And the second part of that, how long after the trial ends will the last mosquito die, right? Will the last Oxitec mosquito die? So how long is it? And how long afterward will our boys be in the field?
Okay, so the first part of this project, which is the single point releases, as I've said, is for about 12 weeks. And then the second phase, which is those neighborhood-wide releases, will be about 16 weeks. So that would take us through to the end of the mosquito season, which is kind of October, November this year. And the second part of that question, how long will these mosquitoes actually survive in the environment? So an individual mosquito is not going to live for very long. It'll live for a few days at most once it comes out. But because these male mosquitoes can pass on these genes to their offspring, and the male offspring will survive and can pass on those genes to some of their offspring, you will potentially see some of these mosquitoes that carry these genes, the fluorescent gene and the self-limiting gene, present in the environment for a few weeks or maybe even a couple of months after the end of the project. So when we've done this in Brazil, we've seen that within about 4 months after the end of mosquito releases, we no longer see any of these self-limiting mosquitoes present in the environment. And we'd expect to see something similar in the Florida Keys. That will depend a little bit on the weather. It'll depend a little bit on how cold it gets and how quickly. But we would expect that that would be a case of a few weeks, maybe a few months at most, before they're completely gone.
Thank you. Andrea, did you want to add to that?
No, I think Nathan covered it. Our goal is to complete both of these projects within this mosquito season. So we will endeavor to do our best to get it all completed by the end of October, early November.
Thank you. So kind of taking it along those lines, we talked about a Project A and Project B. So there's interest on when might Project B begin, and will it be in same sites or new and more sites?
So we don't have a start date in mind exactly for that yet. Our focus right now is very much on just getting Project A up and running. But we would expect that Project B is likely to be in different sites to Project A. And again, Project B has replications, so there would be 3 or 6 sites where we actually do the same thing in multiple locations to be able to understand exactly what's happening and compare that with control sites as well.
Thank you. Some of these questions are they kind of recapitulate. How do you know if the Oxitec males have actually mated with local invasive Aedes aegypti females?
Andrew, do you want to take that one, or I can take it? I don't mind.
No, go ahead.
Okay, so the Oxitec males carry 2 genes. They carry the gene which kills their female offspring, but they also carry a fluorescent marker gene. So what that means is that we can actually look at those offspring and we can stick them under a microscope and they will fluoresce under the microscope. And so we can tell them apart from the wild mosquitoes. So what we'll be doing is we'll be collecting those egg traps that we talked about, bringing the eggs back to the lab, and then hatching them and then analyzing those larvae to see whether or not they carry that fluorescent gene as well. And so if we're seeing those fluorescent mosquitoes, that tells us that a wild female that laid those has mated with an Oxitec male. And so then what we can do is we can look over an area around those release points and see how effectively are we actually covering that area. Are we seeing those fluorescent larvae up to 50 meters, up to 100 meters? Probably not much further than that, but that's the way we would actually be analyzing that.
Good. Couple questions specifically for Andrea regarding Aedes aegypti. And that is, let's see, do the males mate more than once, and are the males larger than females?
So, yes, wild Aedes aegypti males are known to mate more than once, but their lifespan is still very small, about a week probably at best, if he is very fit. And then as far as size goes, depending upon diet for the females, you can really see size variation in females. Males are usually about the same size, if not a tad bit smaller. But what really gives away a male mosquito is very fuzzy antenna. So if you're looking at a mosquito that you've collected and looking at its head, it basically looks like two feathers coming right off the top of its head. And that really will designate whether or not you've got a male.
And the female will have a very strong proboscis, which is designed to try and bite you. So you'll see those as well.
Thank you both. Let's see. You did mention this earlier, but the question has come back, so maybe this is a chance to clarify. How many males come from each box?
So that's a good question. So we would expect to see around 1,000 adult males coming out of one of these boxes. And as I mentioned as well, we're not just going to have a single box, but we will actually be cycling these boxes. So the first box will be placed, and then those mosquitoes will start to emerge after a couple of weeks. But in the meantime, we'll put a second box there as well, so that when the first one finishes, the second one is just kind of getting going. And then the third one will come after that when the second one finishes, and so on, so that we have a continuous release of these male mosquitoes throughout this particular project. But in Project A, throughout all of the release sites combined, we would expect to see about 12,000 male mosquitoes per week actually emerging from these boxes.
Thank you. I was told that there is some feedback when my mic is not muted, so I'm kind of jumping back and forth. How long until the wild female population begins to stop mating with the 5034 males, which is what often happens with sterile release programs? So how long until the wild female population begins to stop mating with OX 5034?
So,
I'm not sure I fully understand the question.
I suppose there's two ways you could interpret that question. One is how long before we start to see an impact on the wild mosquito numbers. So in Brazil, where we've released these mosquitoes, more than 90%, but those releases were done over small neighborhood areas, so a little bit more like Project B. The other way you could interpret that question is there any chance that the wild mosquitoes will somehow figure out that the Oxitec male is putting their offspring at risk and then choose not to mate with them? That's not something which has ever been observed with these mosquitoes. Again, in Brazil, with releases of this particular strain for more than 2 years, we've never observed that. And in releases of more than 1 billion of Oxitec's first generation mosquito in Brazil and other places, we've also never observed that happening. That's something called assortative mating, and it's extremely rare in this kind of situation. So it's not something that we would expect to see, and not something that we have ever seen either.
Thank you. Andrea, some general questions about Aedes aegypti, the invasive species that we are targeting. How did Aedes aegypti get such a strong foothold in the United States? Where did it come from originally?
So Aedes aegypti originated in Africa and most likely came across to the Americas via slave trade ships. So we've seen it established here for a few hundred years at this point. And I think the way that it really established itself is that, it fills a niche that we humans have created. And it has all along. It lives in and around our homes, and it can produce larvae in a small amount of water. What we've seen, a mosquito larva of an Aedes aegypti in a bottle cap, so very, very small, minute amounts of water standing in artificial containers can create issues, which makes it so difficult to control. It's nearly impossible to find every single water source that is out there. We've tried. We ask people to walk around their property, dump out standing water, but it doesn't make a huge impact, unfortunately, on this species particularly. So, they're specialists. They want to feed on humans, and that's what makes them such a great disease vector. That's why they're responsible for transmitting so many diseases, because they like to land on humans, take a small blood meal, not a full blood meal that you might see with other mosquitoes, and then go right to the next person. So they're very flighty, no pun intended. But if you go to try to swat one, it's very difficult. They see you coming and they fly away before usually you can smash them. So they are great disease vectors, and it's really why we're targeting them so much with our control methods.
Thank you, Andrea. The next question is for Nathan. What kind of specific tool will be utilized in identifying the OX5034 with the fluorescent markers? So it's a technical question.
Yeah, so that's a good question. What we will use is microscopes that allow us to look at these larvae in detail, and those microscopes have specific fluorescent light and filters which help us to actually detect the particular wavelength of fluorescence that these mosquitoes will emit. So you do need some specialized tools to actually be able to detect these things effectively.
Thank you. Another rather technical question. Nathan, you did refer to two genes, yeah, the self-limiting gene and the red fluorescent protein. Do you expect both genes to appear in offspring together, or will some offspring inherit one gene and some inherit the other?
Yeah, that's a really good question. So these genes are linked. They are right next to each other in the DNA of the mosquito, so they will be inherited together. You will always inherit the fluorescent marker gene together with the self-limiting gene. And that's something which we've done quite a lot of experiments to demonstrate, and that's something which the EPA assessed as part of the risk assessment process here. So we'd always expect that those two genes will be inherited together. And also part of our ongoing quality control program as we produce these mosquitoes for release is also continuing to make sure that the genes are behaving exactly as planned. So we're always checking to make sure that these genes are fluorescing and that they're killing the female offspring as planned.
Thank you. Andrea, you did describe Aedes aegypti biting behavior briefly. You did say that it tends to feed preferentially on humans. So we clearly have a Keys resident asking, can you compare the biting behavior of Aedes aegypti to the more common salt marsh mosquito, which is so prevalent in the Keys?
That's a great question. I love talking about all of the different species we have here and their different behaviors because they are so different. What I like to say is when it comes to the salt marsh mosquito, they are voracious. You're going to know that salt marsh mosquito is coming after you. And interestingly, the younger they are, so more freshly hatched out adults, are going to come right for your head, right for your face, and higher. And we've seen that the older they get, the lower they remain on the body, which I find fascinating. But that's the biggest difference you're going to see. Salt marsh mosquitoes, again, are very voracious. They're going to come after you. They're not going to leave you alone. Aedes aegypti, many times, I don't even know that they're flying around me when I'm on my porch. They will hide under the chair and come out, take a little sip, and then go back, and then maybe move to the next person. So that's the biggest difference. You're not going to have swarms of Aedes aegypti attacking you all at once. And hopefully, if we do our job as well as we possibly can, you won't see that with salt marsh mosquitoes either. But they definitely are noticeably different behaviors.
I think you anticipated the next question, which is, do these mosquitoes typically swarm? So good, you got that one even
Right.
before we
Well,
asked it.
I think the number too of the population when it comes to Aedes aegypti is very low. You're not going to see large numbers of these all together. Whereas salt marsh mosquitoes, we do have a number of them here. And not only that, they have a synchronous hatch. So you're going to have a lot of females coming off of a swamp all at once. Whereas Aedes aegypti could be a little different. You know, their egg may not hatch upon the first wetting, so you're not going to see large numbers of these moving around together like you do with salt marsh mosquitoes. I think one of the craziest stories I've ever heard is back before, we had a lot of mosquito control down here, is you would see salt marsh migrations picked up like on radar moving from the Everglades into the Keys. So we've not seen that in a very long time, but that just kind of gives you an idea of the numbers that we're talking about and how they migrate long distances together.
I think that's an important distinction as well between the salt marsh mosquito and Aedes aegypti, namely flight radius. As you both mentioned, the flight radius of Aedes aegypti is fairly tight around human habitations. So about 50 to 100 meters from a person's home, very often less than 10 meters from where they were born, about 30 feet. Whereas the salt marsh mosquito can travel for 20, 30 miles in search of a blood meal. To that point, sticking with the biology and kind of comparing mosquito to mosquito, what is the typical lifespan of an Oxitec mosquito versus an Aedes aegypti that you would find in your home?
Yeah, so good question. So in the laboratory, the lifespan of an Oxitec male mosquito is a bit shorter than a wild type comparator. And we would expect to see that in the field, the lifespans are going to be even shorter than what we can actually measure in the lab. And that's what we've seen in Brazil as well. So like Andrea was saying, a very healthy male mosquito in the field might live for up to a week. And that's if it's done really well. And we've seen the same for our male mosquitoes in Brazil. They'll typically live for just a couple of days after they actually emerge. And during that time, they really need to focus on finding a female to mate with. They're not going to live much longer than that time. I actually had a follow-up question just for Andrea, the last one. I was just wondering if, Andrea, you could maybe just speak a little bit more to the other species which are present in the Keys besides those two, the salt marsh and the Aedes aegypti, just to kind of flesh that out.
So you're letting me geek out on all of the mosquitoes, which I love. Thank you. But I think one of the other main mosquito that we see in the Florida Keys is called Culex quinquefasciatus, which is the southern house mosquito. And this is the other one that we do see around people's homes quite frequently, but we find them in more organic water. So swimming pools that are not taken care of, hot tubs, usually larger bodies of water, but definitely very organic. Drains, roadside ditches sometimes, but that's another species that we see a lot of down here in the Keys. And it's really those are the three main ones. But we do have about 45, a little more than 45 species that we find, and each one has its own special little niche that we have to figure out and target. And I think that's why our surveillance program is so important, or any mosquito control surveillance program is so important, because we have to know what's out there, what mosquito is out there, where does it live, and what are the effects that it may have on the population.
Thank you, Andrea. That makes at least three of us in the group that geek out about mosquito biology. A question from a concerned neighbor. We are proposing to, of course, release male mosquitoes. And so a question here, do the male mosquitoes make noise when they fly by your ear?
I think that's one for the mosquito biologist in the room. Again, Andrea, maybe you want to comment on that.
Yes, so the buzzing noise that you hear are wing beats. So there is noise when a male mosquito flies by your ear as well. But, again, the good news is they're not interested in you. He may come near you because a female is close to you. So you should not see any sort of swarming or issues. But if a male mosquito flies by your ear, yes, you will hear it as well.
Thank you. And actually, kind of staying on that same topic, since we will have male mosquitoes. Someone asked, have we ever experienced or documented someone in Brazil having an allergic reaction to these mosquitoes? Not necessarily from biting, but from exposure.
Mm-hmm. The answer is no. So we've released more than 20 million of these OX5034 male mosquitoes in Brazil, and there's never been any kind of adverse reaction. And we've released, as I said, about 1 billion of our first generation mosquito, which had very similar genes and produced the same proteins. The self-limiting gene and the fluorescent marker gene produced the same proteins. And again, no adverse effects on humans, no allergic reactions or anything like that either.
All these conversations, they've been about mosquitoes biting us, whether it's Aedes aegypti or Aedes taeniorhynchus. People know that only the female mosquito bites, only the female mosquito feeds on blood. So this question turns the tables on the mosquito, and someone wants to know what would happen if someone ate one of the OX5034 mosquitoes. So maybe let's expand it and kind of make it a little more relevant, So fine, what if a human ate a male mosquito or your dog or your cat? Because as we know, cats and dogs tend to bite at flying things.
If you ate a male mosquito, whether it was an Oxitec male mosquito or a wild one, you would digest it like you would any other insect that you'd eat. So there's really no adverse effects, whether that was a human or any other animal eating that. And that's, again, something that was looked at in quite a lot of detail by the EPA in their risk assessment. The proteins which have been inserted into these mosquitoes would be digested just like any other protein. And there were also experiments done by independent third-party labs, specifically looking at what about mosquito predators in the environment. So a fish, for example, which might feed on a mosquito larva, or some other kind of invertebrate which might feed on a mosquito larva. Is there any difference if they eat an Oxitec male larva or a wild one? And there was absolutely no difference. They were exactly the same. There was no health impact. They were just as nutritious and calorific as the wild mosquito would be as well. So absolutely no concern there.
I think that's my new favorite word tonight, calorific. Question regarding the size of the Oxitec males. Can you maybe put it in perspective? I mean, most people don't realize when they say mosquito that we're talking about 3,500 species of mosquitoes worldwide, right? We're only talking about one species, Aedes aegypti. And so people have had experiences with, of course, many different species. Can you put in context about how big is a female Aedes aegypti, and about how big is a male Aedes aegypti, and can you compare our boys to the male Aedes aegypti that are already out there? Can you see them with the naked eye, for example?
Andrew, maybe you want to take the first part of that question?
Sure. So Aedes aegypti are maybe about, a centimeter or so. They're not the largest mosquito out there by any means, but they're not the smallest. And you definitely will see them. When you think of a mosquito, that's about, I'm sure most people that are down here know what a salt marsh mosquito looks like. Aedes aegypti are very similar size to that. So maybe, your pinky fingernail, if you trim it really close. And you don't see a lot of size difference, like I said earlier, between the females and the males, especially when it comes to Aedes aegypti.
And just to follow up from that, so in terms of differences between the Oxitec males and the wild ones, really no discernible difference if you were just looking at that with the naked eye.
Thank you. Maybe a question for both parties. Are FKMCD and Oxitec planning more projects in Florida after Project A and Project B? In other words, what's next?
So I can go first, Nathan, and then you can expound. All that the Florida Keys Mosquito Control has planned at this time are projects A and B. From there, we will do a rigorous data analysis involving both our state regulatory and federal regulatory agencies. And with their input, we'll probably figure out what the next steps are going to be, whether anything occurs in the Florida Keys as next on the list or in other areas. I think will be up to those regulatory authorities and Oxitec.
Yeah, absolutely agree. Nothing really further to add there. At the moment, our focus is very much just on completing these projects successfully.
Thank you. A question from a fellow geek. What kind of statistics are you using for this study?
The way that we're looking at the data here are really looking at a few different endpoints that I've talked about. So we're looking at things like longevity, where we might use Kaplan-Meier type models to look at survival. We would be using, again, some kind of linear modeling to look at the distribution of these mosquitoes over the areas where we're releasing them. And then it's just very simple statistics just to compare really the control sites with the release sites, any differences that we might see in terms of things like mating fraction and so on. But I speak as a non-statistician.
Great. I'm actually seeing that I'm not showing up any new questions, which is not terrible. That we're coming up to the hour. I'd like to remind everyone who's watching that if you do have questions that that I have not gotten to, or that you haven't gotten to, please send them to us and we will answer them. Again, if we don't get your question tonight, you can email us at florida@oxitec.com and we'll answer your question if it isn't included in tonight. I'd like to take this opportunity to say that we will have a 14th webinar next month, topic TBD. I suspect we're going to provide updates because this week is monumental, right? This week is really the beginning of a project that has taken 10 years in preparation. And so what we really want to generate from this are the data necessary to proceed to Project B. So we don't have, yep.
If I may also, I'd like to also direct people to visit keysmosquitoproject.com. I know that we have installed a project ticker at the very top to make sure people see any new updates immediately as soon as you go to that website. So you don't have to wait till next month. I know that everyone's sitting on the edge of their seats wanting to hear from myself and Rajiv and Nathan as much as possible, but anytime anybody has any questions or would like updates, it's an excellent resource, and we're going to make sure that it's updated on a regular basis.
I should have mentioned that website. Thank you, Andrea. keysmosquitoproject.com for regular updates. No more questions coming through. If you do have any questions, please submit them. You can get regular updates at keysmosquitoproject.com, and we expect to share our results with you next month at the 14th webinar. Thank you, Andrea. Thank you, Nathan.
Thanks very much.
Thank you.
Thanks for everyone who attended. We look forward to continue engaging with you and with the rest of the community.